Today we're going to talk about algae, and algae are very unusual, which is why we saved them until the end of the semester. Now when it comes to algae, we know that we're dealing with organisms that like to live in water. So for most of the semester we've dealt with land plants. We haven't really talked about the organisms that live in water that are doing some form of photosynthesis, using sunlight energy, and making some kind of carbohydrate, whether that's glucose or whether it's some other molecule. So when we look at this whole grouping of algae, we do want to keep in mind the shared characteristics. So first let's look at the idea that if you're an organism living in water and you're going to be doing some kind of photosynthesis process, what's going to be your main constraint? So when we look back at photosynthesis, remember our inputs were water and sunlight, and our outputs were oxygen and some kind of carbohydrate. Most of the time it's going to be glucose. So we want to first focus on our inputs. If you're living in water, well, you don't really have to worry about that main constraint that all of our land plants had, which was getting water. Okay, so you're always surrounded by water. What's our second input to photosynthesis? That's sunlight. And when we look at aquatic environments, we recognize that sunlight is a major constraint. Let's say we've got a clear body of water. The sunlight can only penetrate so far through that column of water to reach the organisms that need that energy below. What if we have a silty, mucky, real muddy body of water? Well, then our sunlight's not going to penetrate very far at all. So the clarity of water will indicate or will give us the amount of sunlight energy that can reach the organisms that need it. So when we're dealing with aquatic environments, sunlight is going to be a major constraint. The other thing that we do have to think about is algae are going to be categorized into two main groups. One group is going to be the algae that can live in fresh water, and then the other group is going to be the algae that can tolerate salt water. Now, if we have salt water, we have a very hypertonic solution, a solution that has lots of salt, and so there's always that possibility to draw water out of the cells of an organism living in salt water. And so they're going to have metabolic, they're going to have body ways to control at the cellular level, control that loss of water to the surrounding solution. That gets way more complicated than what we're getting into for this class. For this class, we just want to keep in mind that there are freshwater and saltwater organisms, and that the main constraint for all aquatic organisms doing photosynthesis will be sunlight. Now, some of our algae will be doing the formal photosynthesis that we studied in this class, while others are going to be doing a slightly different version, mainly because they don't have a chloroplast organelle. They're going to have phycobilins, and phycobilins will be those pigment molecules that allow the plant to still do some kind of energy harvesting, getting electrons excited with sunlight energy to make some kind of output, some kind of carbohydrate at the end. The other thing that we want to keep in mind is when we're talking about the body of an algal organism, we're going to call that a thallus. The thallus will be a body that could take many different shapes. Some thallus will be just one single cell, while other thallus will be lots of cells bonded together in things like sheets or long hair thread-like filaments, or even more formal body plans that we will look at a little bit later on in this lecture slide. So our algae will be categorized into two broad categories based on the type of cells that they are made of. So some of our algal species will be made of prokaryotic cells. Now, a prokaryotic cell does not have a nucleus, does not have membrane-bound organelles like a Golgi apparatus or chloroplast. So our prokaryotic cells are going to be very different. For this class, we're going to focus in on the cyanobacteria made of those prokaryotic cells. A little bit later in this lecture, we'll talk about the eukaryotic-celled algae. Eukaryotic-celled organisms have a nucleus, have plasma membrane-bound organelles. They tend to be multicellular, but as we'll learn, not all of them, not all the algae that are eukaryotic-celled will be multicellular. And so I think most of the time when we commonly think of algae. We commonly think of these eukaryotic celled organisms. But we do want to start with the cyanobacteria, those algae that are prokaryotic cells, because a lot of times they are the ones that we eat and can also cause a lot of hassle and problems in our lives. So let's dive into the cyanobacteria. Here we're seeing a picture of spirulina, and I love this picture because finally we can see why scientists named this algae the way that they did. That spiral shape of the individual cells and the filaments in that thallus are all very, very spirally and help us to understand how it got its name. Spirulina is a type of edible cyanobacteria. And I like this photograph because it shows us how nutritional, how much nutrients we get out of just a small amount of spirulina. So spirulina has a lot of really good things and has been eaten by people for a long period of time. Keep in mind that here in the Midwest, we have a slightly different palate. Because we aren't living near the coast where we have fresh saltwater organisms to enjoy, we don't have that continual exposure to what algae might taste like and what organisms from the sea and aquatic environments might taste like. We don't tend to think that things like spirulina taste really delicious. It can be a bit of an acquired taste. But for people who live along the coast who have been eating these types of algae and saltwater organisms for their entire life, they don't notice necessarily that the flavor would be off-putting. So if you are from the Midwest, you may want to ease yourself in if you start to eat algae and know that it might take some time to acquire the taste for eating things like spirulina or even things like nostoc. So here we're looking at a picture of nostoc. Looks a little bit like a necklace. And the important thing about the nostoc is this heterocyst. So we have individual cells that have bonded together into a filament-like thallus, and this heterocyst is going to be a cell that is dedicated to nitrogen fixation. I really like this picture because it's beautiful. We can see those individual cells bonded together, and this great big open heterocyst dedicated to nitrogen fixation. Remember, nitrogen fixation was something that we saw with our land plants. When we covered the legumes, the main characteristic of the legume, or the big important thing of the legumes, was that they could form that relationship with rhizobia bacteria. The rhizobia bacteria would take the nitrogen gas from the atmosphere and turn it into a form that the rest of the plant could eat. Remember when we talked about the grasses and the azolla fern living alongside the rice? The azolla fern was grabbing nitrogen gas from the atmosphere, turning it into a form that the rice could eat. Here we see at the cellular level that same type of relationship. This heterocyst is going to be turning nitrogen into a form that the other cells can eat. And we look at edible nostoc, it has that more traditional kind of slimy look that a lot of us associate with algae. It is a bit of an acquired taste, but it is incredibly nutritious, just like our spirulina. Now, here we're looking at a satellite view of blue-green algae, another one of the cyanobacteria. This big lake up here is Lake Mendota in Madison. Here is the Capitol building on the isthmus, and this is Lake Monona. Connecting the two is the Yahara River. The Yahara River starts up here and helps form both of these lakes flowing through Lake Mendota, and will continue on until it eventually connects to other rivers as part of that larger Mississippi watershed. The reason why I wanted to show you this satellite image is for this beautiful green striation and these big patches of green in the lakes. Those are the blue-green algae blooms, and these are incredibly, incredibly toxic. Because there's so many people researching these lakes, what they did many years ago is they tagged the individual molecules of nitrogen and phosphorus that were being put on the farm fields way upstream, not even being shown in this picture. Then they were able to track those molecules moving using satellite images. And so they got that connection between the fertilizer that farmers were putting on the field, running off into the waterways like the Yahara River, and then ending up in these big lake ecosystems where they then fueled the bloom of algae. All of that nitrogen and phosphorus food just causes the algae to grow like crazy. Now, the big concern when we see this type of algal bloom is that these algae are releasing a neurotoxin. It used to be thought that humans wouldn't have an effect, but unfortunately we now have research that shows us that humans have been having an effect. Usually the effect is going to be nausea, fatigue. If you get chronic exposure, day after day, year after year, you could start to deal with some serious liver damage. So keep that in mind. This type of blue-green algae is incredibly unhealthy in our ecosystems. Now, this is the swimming pier at the Memorial Union at the UW-Madison. It's kind of central to the university. Just up the street here are all of the fraternities along the lake. So you can imagine that this part of the lake gets a lot of people swimming in it in the summer. And the reason why I included this picture is because of all of this slime. This is the blue-green algae bloom. And for many years, I would watch people a little bit tipsy come running off the pier, great big beautiful swan dive or cannonball right into the lake on the hot sunny days, right into this blue-green algae bloom. And of course, later on, they may feel a little bit sick, but they have been drinking all day. So the big question is, was their sickness due to the drinking or was the sickness due to the exposure? We now know that this exposure is contributing to part of the illness. Now, of course, for the animals that can't go anywhere, the fish, the crawdads, the little mussels, they're stuck in this aquatic environment. They can't leave. They have to deal with the neurotoxin, and a lot of times we'll see fish kills and we'll see a lot of dead organisms associated with that neurotoxin. Now, I wanted to show this bigger picture, bigger problem that we're having through our Great Lakes with these blue-green algae blooms. So this is Lake Erie from 2017. And then we also have bigger algae blooms that happened in 2018 and 2019. And what this blue-green algae bloom looked like up close was really disgusting. When we take a step back, we have Lake Erie down here, and we can see how concentrated that algae bloom was. Again, all of the runoff from the fields, from the yards with the nitrogen and phosphorus fertilizer that doesn't make it to the plants, it just ends up in the aquatic ecosystems, causing these algae blooms in our Great Lakes. Now, the reason why I wanted to step out and take this bigger look is because we're starting to see some of those algae blooms in our other Great Lakes, especially along around the areas that are highly concentrated with human activity. This is what it looks like up front. Look at that slimy green, just how many individual algae there are. And then, of course, this is incredibly, incredibly toxic water with all of these individual cells secreting neurotoxins. Now, unfortunately, over the last few years, what we're seeing is an increase in pet death, and it happens really quickly. So if you're going for a walk and you're by the lake and you let your dogs run in, you think, it'll just be a quick second. They'll jump in, swim around, you yell at them, come back here, you're going to smell disgusting. By that time, they would have already been exposed to so many neurotoxins. One woman reported taking her dogs to the lake. They jumped in. They were in for just a few seconds. And on the car ride home, they started to have seizures. Before she could even get them to a vet or even understand what was happening, they had already died. So please, 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 if you have pets, and even for yourself and your friends and family, don't let them swim when there is a notification of blue-green algae growth. Don't let them swim if you can see the blue-green algae. We know that this neurotoxin is very damaging to humans. So that wraps up our sort of brief look at all of the algae that are classified in the domain bacteria, those mostly single-celled organisms that are causing all kinds of different situations. Some are edible, some are problematic. Now let's switch gears and look at the algae that are classified in the domain eukarya. So our first domain that we or kingdom that we want to look at are the kingdom of protists and this division Pyrophyta. Now, Pyrophyta refers to flame-colored, and some of these can make a very dramatic coloration. Most of the species in this division have a single-celled thallus. And what I like about this picture is that it shows us how geometric those thalluses are, those individual single-celled are. So they tend to be very geometric, and then they also have a flagellum that they're using for movement. And what I find just amazing about these individuals is when we look at something like this, the way how it's going to move is by spinning or wiggling that flagellum through the water. In other words, we're not going to get straight line movement. They're going to be able to go in all kinds of directions. And for that individual single cell, I always wonder sometimes if it feels like, I wish I could just go in a straight line. Because attaching a whip and essentially using that whip to move yourself is a very, very inefficient and very strange way to move through the aquatic environment. But there you go, shared characteristics of some of the individuals in the division Pyrophyta. Here we're seeing some of those very geometric shapes. And the individual species in this division some of them can do bioluminescence, that ability to produce their own light. Their own light looks a little blue, a little spooky, at least to our eyes. Now, from satellite images, we do have satellite images of the Earth at night, and over the oceans you can see these great big blue streaks of all of those individual single-celled species luminescing and making that blue light. I wanted to exit out here and play for you this video of somebody kayaking through one of these algal blooms because it's just wild. So they're in a clear kayak. We can see their feet. And all that blue, as the paddle in their hand hits the water, those are individual cells luminescing. Wouldn't this be wild? Take a look at another type of algae. This is the species that causes the red tide. Now, similar to what we saw with the cyanobacteria in the blue-green algae in freshwater, Lake Mendota, Lake Monona up in Madison, Wisconsin, here we have a saltwater species in the domain Eukarya doing that same similar thing of secreting a bunch of neurotoxins, and those neurotoxins are going to be killing a lot of the fish in the area. Now we see these big blooms of this red flame-colored algal species whenever we have a lot of fertilizer, a lot of phosphorus, and nitrogen. So a similar cause as to why we get these huge explosions in population numbers, these huge blooms of individual single-celled organisms that then secrete a lot of neurotoxins that can lead to huge fish kills. Now here we're seeing a fish kill from a very different species. This is the Carina brevis dinoflagellate. And in the Gulf of Mexico, when we get huge blooms, for a similar reason, lots of fertilizer in the area means the algae is going to grow. So one of the things that tips us off that there might be a big algae bloom if we haven't physically seen it is we get these huge fish kills washed up on shore. Now, if you can just imagine walking through the woods and smelling this and the intensity of the smell of all of these poor dead individual fish. When we see these big fish kills, we do have to think about the rest of the ecosystem. Remember, the little fish get eaten by the bigger fish, get eaten by the bigger fish, etc. We know that this is a huge ecosystem disruptor. And a lot of times the things that can happen is we can end up with irreparable damage. We can't get the ecosystem healthy again. Once we've seen this explosion in one species of algae and the neurotoxins and all of the accumulated problems, it's easier to just not have the big explosion of algae in the first place. Now, everywhere we have human activity, everywhere around the planet, not just on the coasts, we have these huge algae blooms, we have the toxins, we see this ecosystem disruption. Oh, I don't know what to tell you about this. The problem is fixable. We know we can fix this, but we have to have the concerted effort to want to fix this. Otherwise, we're just going to have to accept these really unfortunate circumstances, the toxins, the smells, and all of the other issues and problems associated, including things like Pfiesteria that causes these really gross and awful lesions in fish. Let's change gears a little bit. Let's get into the division Chrysophyta. Again, we're still in the domain Eukarya, so we have those algae that have plasma membrane-bound organelles and have a nucleus. Now, these individual species have a lot of unusual cell shapes. And one of the things that these single-celled organisms will do is they will take and incorporate silica, essentially making glass cell walls. Now, that sounds kind of fragile. Like glass doesn't necessarily sound really stable to us, but in the aquatic environment, this works out very well. And of course, I love the diatoms because diatoms can be used to make diatomaceous earth. This is a natural insecticide. Remember when we talked about the starchy staples and I talked about the potato beetle larva, that soft-bodied insect? If you put this dust out there, and this is what it looks like underneath a scanning electron microscope, you can see all of these shards, and you can see this aggressive shape that the diatom's cell walls will break into once we take their cell walls. The cell is dead, but once we take those cell walls and crush them up. This is also why diatomaceous earth, as a nice white powder, can be so dangerous to inhale. So you got to wear a mask whenever you're spreading it. That way you don't get any of these little glass shards done into your lungs where they can cause some damage and a little bit of irritation. But if you can imagine a caterpillar, a larva, crawling across these shards and just scraping up their body. Now the drawback is that anytime it gets wet, the diatomaceous earth is not as effective. So just to put it in perspective, remember these are single-celled organisms. The diatoms, when they die and fall to the bottom of a body of water, will accumulate. And so here we have the mining of diatomaceous earth. There are just that many single-celled organisms building up to that immense thickness at the bottom of the body of water. The division of euglenoids is really fun. And the reason why I think a lot of people enjoy the euglenoids is that they're very active. So when we look at them in lab underneath the microscope, these are cells that are constantly moving and they can move really, really quickly. Now, hopefully, if you look at them underneath the microscope, you can see the little red eye spot. Most of the time, what we see with these individual organisms is that they're mostly clear on the inside. We don't underneath, using our microscopes in the lab, we don't necessarily see all of the chloroplast organelles, but sometimes we can get to see one of those little red eye spots. Now, the eye spots are going to be perceiving light and helping to guide this cell where the light intensity is stronger. So here we see more like what we would see underneath a microscope in our class. Maybe we'll get to see that little red eye spot. I hope that you can see it because I think that it's really, really fun. Another division in our domain Eukarya will be the Chlorophyta. These are formerly known as green algae, but don't think of it as every time you see a thallus that's green, it's automatically classified in the Chlorophyta division. That's a little bit of a misnomer. This is where I get frustrated with our common names. With our shared characteristics, we find that with our green algae, our Chlorophyta, we are finally going to have thalluses that can be multicellular. The Chlorophyta algae are often found in that symbiotic relationship with fungi called a lichen. They can do sexual and asexual reproduction. So here we see a single-celled species, the Chlamydomonas, Volvox, that hopefully will be living when we see in labs. Sometimes they don't always survive very well through shipping and handling. these are going to be multicellular. And what I love about volvox is they look like little soccer balls. Each of the cells on the outside of our three-dimensional spherical volvox will have the dedicated flagella. In other words, their one role is keeping our little soccer ball of cells spinning and rolling through the aquatic environment. These kind of darker dots inside, those are actually lots of cells together. These are daughter colonies getting ready to bud off from this one larger volvox colony. So here we see multiple cells coming together in a spherical three-dimensional thallus, and we see that cell specialization, and that's really unique and unusual. Some of our larger multicellular chlorophyta algae will have true blades. Now remember, this can't be a leaf because we don't have that dedicated vascular tissue, xylem and phloem. We don't have the same type of dermal and ground tissue. These are plants living in water, so we're going to have a lot of different types of tissue. We'll have the blade, and we'll have a stipe that's kind of stem-like. The blade and the stipe together create something called an entire frond. Now sometimes our fronds are going to have little gas platters. These are dedicated packages of cells that blow up and look like a bubble, trapping air inside. It's going to help to hold that frond upright in the water column so that when this algae thallus is experiencing the flows and fluxes of tides and currents and the way how water can move, that frond with the blade is always being kept as upright as possible and as close to the sunlight as it possibly can be. Some of our algae are going to have a holdfast. Those root-like structures are going to keep them anchored in one spot. Again, when you're constantly being moved by the water, you want to stay where you are at. You don't want to be constantly floating, potentially to a place where there isn't any sunlight. So here we're looking at a very common species in the division Rhodophyta. Rhodophyta refers to red algae, and we can see on this frond we've got some red coloration, but then notice how some of our frond is also green as well. So don't let the color necessarily dictate what the algae is classified as. Most of our species in the division Rhodophyta will have that multicellular thallus. And here's some classic multicellular thallus fronds, really great big flat sheets, and some of those more feathery type fronds that we associate with algae. Again, some more fronds. Note the frond of the chondrous algae up here. There's going to be some lab readings about the chondrous red algae. Some of the edible species from the division Rhodophyta will be dulse. And if you notice here, dulse, when it's dried up, looks pretty brown, very nutritious. Nori, that's commonly used in sushi, is also a type of species in the division Rhodophyta. And then here we're looking at E. cottony. Now this is one of the most commonly consumed types of algae. From E. cottony, we are going to make carrageenan. Carrageenan is found in all kinds of things as a thickening agent. So if you look at some of the, like eggnog, or you look at things like soy milk or oat milk, you look at puddings, even ice creams, you will see that carrageenan ends up in these products to help thicken that liquid. So I encourage you to take a look and start reading labels and notice that anytime you see carrageenan, you are eating algae. Now, of course, this is one of the least fishy or seawater tasting type of algal species that we consume. Another type of algal species that you may have consumed or may have even used yourself in cooking is agar. Agar is also used as a thickening agent. In the lab, when we did the lab where we took bacteria and spread it on a petri dish, and in that petri dish we had jelly. That jelly was called agarose gel. So in science, we use algae all the time, especially when we're making that agarose gel for bacteria to grow on. Our next division of multicellular multicellular algae is the division Phaeophyta. Phaeophyta are generally called the brown algae. Here we see some of the common thallus types. Again, we're looking at those fronds that can be very fern-like or can be more sheet-like. And some of the more enjoyable or common types of Phaeophyta brown algae that you will find here in the US will be the rockweed. And rockweed is great because it has all of these really distinct bladders. Now, I will tell you, when you pick up the rockweed and you start to pop those bubbles, it is as addictive as popping bubble wrap. I mean, it has that same kind of explosive kaboom when you pop it. It's really, really quite remarkable. All of these gas bladders are going to keep that frond up near the surface of the water, essentially assuring that this algal species can outcompete other algal species by grabbing all the sunlight. Here we see kombu, and kombu is an edible type of brown algae. And one that we see more commonly around here, I find it all the time at Woodman's, is wakame. I know it's called commonly brown algae, but look at how intensely green that is. The last type of algal species that I would like to talk about is this Caulerpa taxifolia. This is an invasive algal species that is growing out of control in the Mediterranean Sea. It's got very feathery fronds, and it's going to have this kind of rhizome horizontal spreading structure. So what this species can do is it can take over an area very, very quickly. It's going to outcompete the other algal species. And so essentially what we end up with is this huge monoculture of one species. What this does is it kills the ecosystem. We lose all of the diversity. And around the Mediterranean Sea, we have so many people who are deeply dependent upon fishing, upon the creatures, upon the organisms. They depend on this ecosystem, not only for food and sustenance, but also for their livelihood. Ecotourism, snorkeling, all of the reasons why people really want to live and visit this region on our planet. So when we see an area like where this diver is at, where it's all this one species of algae, we are essentially looking at what's almost like a barren landscape. There's so little diversity in it. We don't really have a good solution to this yet, like so many of our invasive species. They're good at outcompeting the organisms that already exist there. We don't have a way to get rid of them. The only thing that we keep saying over and over again is don't let them get there in the first place. But that's very challenging with how much humans love to travel and how much we love to spread things. So, if you're looking for a problem to solve, we've got so many of them when it comes to invasive species on land and also, as we see, in the sea. I didn't want to leave you on just a depressing note, like we've got all these problems that need solving. I wanted to show you how algae are actually helping us solve lots of problems all at the same time. And it falls under this bracket of coupled systems. When we have coupled systems, we have two or more interdependent systems, where the outputs from one system are the inputs for another system. And this is a beautiful example of how we can solve multiple problems by linking our systems together. So in these great big tubes of green stuff, what we have is lots of algae. Algae are doing photosynthesis. Our inputs for photosynthesis are carbon dioxide, water, and sunlight. So if we give these algae lots of carbon dioxide, they're going to make lots of outputs. They're going to make lots of glucose. So what these tubes are doing when they're sitting outside of this great big power plant is the power plant, in this case, is burning coal. When it burns coal, it's going to be making carbon dioxide emissions. Carbon dioxide emissions are food for all of this algae. So we take the carbon dioxide emissions, we run it through the tubes of algae. These algae are going to eat the carbon dioxide. They're going to make lots of glucose. Well, what do you do with all the glucose? Glucose can be an input for making ethanol, for making biodiesel, for making the fuels to run our vehicles. Remember, our vehicles run on the fossil fuel crude oil. That's in limited supply. As the supply of fossil fuels goes down, the cost is going to go up. So we know that we have a problem with our vehicles, how we move people, how we move stuff. The main fuel that we use to run our vehicles is going to increase in cost over time as the supply decreases. So by taking these algae, these single-celled organisms that can eat carbon dioxide and make glucose, we can solve two problems. Grab that carbon dioxide that we know is contributing to climate change and produce glucose, which can offset the use of crude oil in our vehicle fuels. We have two or more interdependent systems where the output from one system is the input for another. The output from the coal plant, carbon dioxide, is the input for algae. The output from algae, glucose, is the input for making ethanol for replacing gasoline in our engines. And since that was a drawing, I didn't want you to think that this was just theoretical. Scientists have been thinking about it, but we don't have a real-world example type of situation. This is a picture of a power plant down in Kentucky. Now, they don't have the full scale yet. This was just a test that has been running for many years. I think we're looking at almost 17 or 18 years of them running carbon dioxide emissions through these tanks of algae and harvesting the glucose to use in other systems. So we know that this can work. We know that we have a solution to multiple really challenging problems. And I can tell you that whenever we do this, whenever we can look at the larger picture, at how everything is interdependent and how the outputs from one can be the inputs for another, we find elegant solutions to really complicated problems. So our algae are weird. Some of them are in the domain bacteria because they're single-celled, prokaryotic celled organisms. Some are in the domain eukarya because they have the nucleus. They are eukaryotic celled organisms. Some are single-celled, some are multi-celled. Some are commonly called red algae or brown algae when they don't even have that color whatsoever. My only tip to you in this chapter is to do your best to memorize and get the associations down as best as possible. We've done just the briefest overview of algae. You could spend a lifetime studying these very interesting, very complex organisms. And of course, it is the algae that give rise to our land plants. So all those plants that we've been studying this semester have their DNA roots back in the algae that once lived in the ocean. Life on this planet started in the ocean before it was able to move onto land. So if you have any questions, as always, please email me.